All right. I want to start us off with a childhood memory. So we're turning the clock back, and back, and back to the night that my mother and I crossed the border. There was no moon to guide our path. There was no wind to cover our tracks. Even the river was silent in anticipation and hesitation as my mother lifted me in a black trash bag and eased her way into the water. Off in the distance, there were lights and voices flickering on the bridge as the cold touch of water wrapped around my feet, my knees, my stomach, my chest. It stopped just before my head. At this point, I couldn't see my mother anymore. The water was too deep. The current kept stealing away the seconds as I waited for her to take a breath. And yet, feeling her warmth through the bag assured me that everything was going to be okay. And we're back. What I just described was a memory from when I was 5 years old. My mother and I crossed the Rio Grande from Mexico to Texas in search for a better life. Surprisingly sharp and vivid memory, right? Especially from so long ago. When we think back to such sharp memories, it can feel like traveling back in time as we just did. And why is that? Why is it that some memories can be recalled so sharply, so clearly and vividly, while others, who are left with keys, are so vague? Well, thanks to our lab's latest discovery, we might be one step closer to understanding why. But first we have to ask the big question. What is memory? We can think of memory as a storehouse for our experiences. We box away the moments we live, and then sometimes lose them as we pile up those boxes. Not everyone is going to have a vivid memory of crossing the river like I do, but there are other memories that can be just as sharp, just as vivid, and many more that are just as boring. Think back to your first love, your grandmother's cookies, the time you twisted your ankle, or preparing for that baby. I'm sure most of us here have experienced these things at different points throughout our lives. But if I were to ask each and every one of you to recount to those memories, I'm sure we'll start to notice how some are glorier than others. This tells us that memory is more than just a storehouse for experiences. There's a lot more to it than that. But our definition and understanding of memory is still incomplete. Which is why we're then wondering things like, what determines whether a memory will be packed away into the storehouse, or if it will become a big, furry mess. Our first and perhaps most obvious suspect is the one responsible for creating the experience of who we are. The brain. Specifically, the brain region responsible for learning and memory. The hippocampus. If we can understand how brain cells of the hippocampus transform our living, breathing experiences through these electrical signals and store them as memories, then maybe, just maybe, we can start coming up with solutions to help those individuals with learning and memory problems. Well, it's here at the hippocampus where we discovered a unique population of cells never before studied that could be playing an important role in how we remember things. Shown here in this beautiful image I captured of the hippocampus are newly discovered cells in gold. And the way we study them is by modifying the DNA of myocyte viruses to turn these cells and only these cells on or off. Out of the billions of cells in the brain, we are the world's first research lab to be able to target and study such a unique population of cells. Now, if you're wondering why we want to turn them on or off, think back to a time when you entered a room with an unfamiliar switch. Does turning the switch to flip the lights on? Is it for the fan? The shutters? Does it even work at all? Only way to find out is to flip it. So, just like that switch, We flip ourselves on, and what we discover is fascinating. How to blur memories. Now, how exactly did we do that? How did we use these modified mice and viruses to turn our special cells on or off and figure that we can blur memories? The way we went about this was by teaching mice to learn a specific association between stimulus and environment. Then observe the reactions anytime they're in that specific environment to tell us that they remember the stimulus. So when we put them in a distinct and unrelated environment, that mice doesn't react because there's nothing there to remind them of the stimulus. Now, when we turn our special cells on, we start seeing a reaction. The mice are remembering the stimulus, even though there's nothing there that would normally remind them of it. Well, I see those confused faces. What does this mean exactly? What's going on here? Well, one way we can try to paint the picture here, at least that I would like to try to paint the picture here, is through another childhood memory. So we're back in time again, rolling in the humid heat of an unfamiliar border town. A patrol cruiser haunts the crowded streets. My mom squeezes my hand to hurry out of sight. I learned that I want to avoid the police because getting caught meant deportation. So anytime I heard the sirens or saw the police, my eyes would go wide. My heart would beat out of my chest. And my lungs would burn as I struggled to keep up with it. This is the learned association between specific stimulus, environment, and reaction, which in my case was too high. Now, imagine being in the comfort of my home, relaxing, when suddenly there's a knock on the door. Knock. Before I go in, I'm hiding underneath the bed. There's nothing but this old thud. My blood pulsing against my head. Weird, right? I reacted as if I'd seen the police out on the streets, despite being in the comfort of my home. This is what we're seeing when we turn our special cells on. We're blurring that learned association between stimulus and environment to such an extent that you trigger a reaction, even in a distinct and unrelated environment. This, combined with more of our data from our lab, is telling us that what we're doing is no longer neatly packing away those memories into the storehouse, and instead blurring them into the present moment. Now, this might still be a little hard to follow, which is completely understandable. Memory is a pretty abstract concept. So we can think about it through a different perspective. In the beginning, I shared a sharp and vivid memory of my mother and I crossing the border. I was terrified that we'd get caught, especially of being swept up by the currents around me. And yet, I grew up to be a swimmer. In fact, it became a very big part of my life. Despite how sharp and how vivid this memory is, it stayed deeply packed in that storehouse. And yet, when we turn our special cells on, that's not the case with our mice. Can we reverse this? Can we turn our special cells off and allow our mice to sit comfortably in their homes without worrying about someone knocking on the door? Could we sharpen memory? Now, our work is still ongoing. There's a lot more to be done, and what we've discovered is only the drop in the ocean of what's left to be discovered. But it's only a matter of time before we can improve our understanding and figure out what determines whether a memory gets neatly packed into that storehouse or becomes a big blurry mess. To finally figure out how our brains turn these electrical signals into memories that they later store. And finally answer that big burning question. What is memory?